आवाज कट होतोय का तुझा जरा?
हो आता ते
हॅलो
आता
हॅलो
ते आवाज व्यवस्थित.
हा
हॅलो
हा बोल की.
येतो ना व्यवस्थित आवाज?
हा ठीक आहे काय
हा
अडचण.
व्यवस्थित आवाज येतो ना सर? ओके.
नाना साहेब प्लीज मेक co-host or speaker.
Sir, I think you are host. You can make
Yes.
host the.
Good morning Aalok ji.
Good morning, sir. Good morning. How are you, sir?
Fine. Welcome.
Thank you, sir. Thank you. Good morning, sir. Good morning, Dahat, sir.
Good morning. Hello, good morning.
Yes,
How are
sir.
you?
How are you, sir? I'm good, sir. How are you, sir?
Okay. Nice to see you.
Yeah, sir. Same here, sir. Same here.
Okay. I thought you will be taking the topic on genetics, so I thought I should also join. But just now I saw it is photosynthesis. Let it be. But
Yes.
I will also enjoy your lecture.
We don't have that topic, sir. Actually,
Okay, anyways.
the general agriculture, we need to cover photosynthesis, respiration, and transpiration topic.
We are your
Just,
students, sir. We have enjoyed those days studying the genetics.
Okay, good. And same I am continuing here also.
Yes, nice.
I request Mr. Nana Sahib, please introduce our today's speaker so as to start the session.
Dear participants, my name is we are with respected and Indian Institute of Sugarcane Research, Lucknow, UP. Very warm welcome, you, sir. Sir has completed his BSc Agriculture program from MPKV Rahuri, MSc in Genetics and Plant Breeding from INA Navsari, Gujarat, and his PhD in Genetics and Plant Breeding is ongoing.
Sorry, sir, but your voice is breaking in between.
Yes.
We are not able to listen you properly.
I think he left. On behalf of organizing committee PGI MPKV Rahuri, Department of Agriculture Extension and Communication, I am very much thankful to sir that is he is present with us and deliver a talk on particularly photosynthesis, respiration, and different techniques of physiology particularly. I request Mr. Aalok sir to please take over the charge and complete and start your session, please.
Sir, the students has joined or they are yet to join?
Yet I just mail all the students.
Okay.
They will join within a short time.
Okay, so should I proceed or?
Yes. Please, you proceed.
Okay, so a very good morning to all. And first of all, let me thank you, Dr. D.V. Dahat sir, Dr. Prasant Patil sir, and all the organizing committee who has, planned for this wonderful lecture series. And I hope it will be really helpful those preparing for the JRF, SRF, and ARS. So the topic which is given to me is very basic, and so I feel like it would be a revision for all. I will try to keep it as simple as possible, and so that the people who are listeners will take a short and sweet message from my lecture. So let me share my screen. Sir, I'm not able to share my screen because it is saying host disabled participant screen sharing.
Dr. Prasad,
I have make you co-host. You please make changes. Nana Sahib.
yes, sir. Now you can see it.
Please share your screen, sir.
Okay. Still, again, this message is coming host disabled participant screen sharing. Is it like I have to relog in again here?
No need.
No need.
Dr. Pawar, please see.
Yes, please.
I think Vidya Bharati is host now. Ask him to make the
Like that.
Ah.
Wait, I will. This is one of the problem of online sessions.
It's okay, sir. No issue.
Vidya Bharti, are you hearing me? Hello. Hello.
He has muted himself also.
Hello. Vidya Bharti, are you there?
Mhm.
Wait, sir, I will log in again.
Okay, sir.
How is everything going on, Aalok?
Yes, sir. Yeah,
How is going
everything
on
is,
there?
yeah, everything is well. Actually, I'm involved here in the main varietal development, sir.
Okay.
So I'm seeing all the ACRIP trial along with the main PI, Dr. Sanjeev, sir, and
Okay.
other scientists. So
You don't come
mainly
here
we are.
to Padegaon or so?
No, actually, Padegaon is one of the center, voluntary center
Okay.
in ACRIP, sir. So maybe we could visit in future there, sir.
Okay, then do come. I am near to Padegaon only.
Oh, that's nice, sir. Sure, sir. Definitely, I'll come to meet you.
Hardly, I think 50 or 60 kilometers away from Padegaon.
Okay, sir. Okay, sir.
Or
Sure.
you don't have that. Also in Kolhapur, ACRIPS trials are there. In Kolhapur also. Mostly of
Yes.
Jaggery is there. PhD
Aalok,
on
sir,
Jaggery.
you are host now. Please share.
Okay.
Aalok, continue.
Okay, sir. Thank you. Uh, is my slide visible?
Yes. Please continue.
So once again, I welcome you all in this e-training courses on ICR, GRF, SRF net, and ARS examination. Myself, Aalok Shiv, and I'm working as a scientist in Indian Institute of Sugarcane Research, Lucknow. And in this lecture, I'll be talking about the photosynthesis, respiration, the factors affecting photosynthesis, and in very brief, what is photorespiration. So as we all are aware that what is photosynthesis, so photosynthesis is nothing but the process which converts the solar energy into the chemical energy, and that is used by the biological system, that is for us. So here we can see in the presence of sunlight, this energy in the plant with the help of chloroplast, the carbon dioxide and the water is utilized and it is converted into the sugar, and it is all the byproduct of this process is also the oxygen and the water. So in photosynthesis major there are three events. First, the sunlight is converted into the chemical energy. Chemical energy is nothing but the glucose, and the water is split into the oxygen. The water utilized is liberated in the form of byproduct oxygen, and the carbon dioxide is get fixed into the sugar, that is glucose, the simplest form. So here we can see the reaction of photosynthesis. Here the carbon dioxide and the water is utilized in the presence of sunlight, and we are getting the glucose molecule, and along with that water is released. So photosynthesis is not only carried out by the plant, it is also carried in certain bacteria and phytoplankton and most of the algae. These are called as a photoautotrophs. Autotrophs means those who can prepare their own food, and photo means in the presence of light. So those who can prepare their own food in the presence of light, they are called as the phototrophs, and they are actual producer of in this system. And the consumers such as herbivores and carnivores depend on these producers for these. So why glucose? Why do plant make the glucose? Not on why not other carbohydrates? So there are certain reasons for this. The glucose molecule can be broken apart for energy to power reaction. So with the help of this, glucose can be further break down to provide the energy. It is the smallest and it is the simple sugar and the smallest unit of the carbohydrate, which could be converted into the other polysaccharide like cellulose is starch, which could further utilized. So this is the reason that plant make the simple and the smallest unit of carbohydrate, that is glucose. So where does this process takes place? So this process in the plant takes place in the chloroplast. So here we can see the structure of the chloroplast. So in this chloroplast, there is a space called stroma, and this is the place inside the chloroplast. And these are the thylakoid. These are the round flatted disc. This single disc is called as a thylakoid, and the stack of these thylakoid is called the granum. So different process or different part of the photosynthesis takes place in the different part of the chloroplast. So if you'll see the leaf, and through the leaf section, we can have the plant cell. In the plant, we will find the chloroplast. In the chloroplast, we'll have the granum. And if you'll see the granum, it will have the in thylakoid. There are different pigmentation system and different proteins which help in the photosynthesis. So this is the structure where we can see the different enzymes like ATP synthetase and other enzymes situated in the thylakoid membrane, which help in the formulation of the ATP. So photosynthesis is mainly divided into two parts: the light reaction and the dark reaction. The light reaction is mainly because the light it is dependent on the light. So this takes place in the thylakoid, and where in the pigment thylakoid membrane protein, and there are different protein presents in these thylakoid membrane, and they form the photosystem first and photosystem 2. And these photosystem harvest the photon energy that is obtained from the sun, and they convert them into the energy molecule. And if you see the dark reaction, it is independent of the light and do not need the light energy to power their reaction, and can occur either in the day and light. It is discovered by Kelvin Benson. It is also called as the Kelvin Benson Ambassador cycle, or just Kelvin cycle, because of the scientist who has discovered. We'll see now the light reaction in detail. So in the light reaction, what happens? The energy absorbed by the chlorophyll during the light reaction is used to power the photosystem second, that breaks the bond of water absorbed during the absorbed through the plant. So here we can see the water which is absorbed through the plant root is broke down, and this oxygen is released through the stomata, And after this oxygen is released, there is leftover is hydrogen ion, that is atom. So what these atoms do, these atoms, when they are in abundance, they move out of this thylakoid membrane, and when they move, the ATP maker, that is ATP synthase, these enzyme, with the help of ADP, it convert them into the ATP, and ATP is the source of energy. So this is how. And also in this, with the help of photosystems too, this ADP converted into the ATP, but also in with the help of photosystem, first this NADP converted into the NADPH, and this is also energy currency for the plant. And this NADP then carries the energy over power to the dark shades. And this ATP and NADPH, which is produced during the light reaction, is now utilized by the utilized in the Kelvin cycle or the dark reaction. So in short, if we'll see the light reaction summary, the photons are absorbed by the pigments to power the photosystem first and photosystem second. So we have seen in photosystem second, these protons are absorbed by the ADP and adenosine diphosphate, and they convert the ADP into the ATP with the help of enzyme ATP synthetase. And with the help of photosystem 1, this NADP converted into the NADPH, and these ATP and NADPH utilized by the dark reaction. So this is in short. Here we can see the oxygen is released. Now with the help of this H molecule, there will be a formation of ATP and NADPH. Next is dark reaction. Dark reaction is also called as the Kelvin cycle because of the scientists who discovered, and it takes place in the stroma of the chloroplast. We have seen the different parts of the chloroplast, so it happens, the light reaction takes place in the thylakoid, and the dark reaction takes place in the stroma. So what happens in this Kelvin cycle? This Kelvin cycle starts with the RUVP, rubisco biphosphate molecule, and the carbon dioxide, and it requires an enzyme called rubisco. So what this rubisco do? Rubisco, this RUBP combined with the carbon dioxide in the presence of enzyme rubisco, and it form a intermediate compound which is unstable. So what happened? This enzyme is unstable. This product is unstable. So it get split into the two stable product, that is 3-PG, phosphoglyceric acid, and these two products, with the help of ATP and NADPH, which is produced in the light reaction, they gets converted into the G3P, and this is the final form. And the ADP and the phosphate and NADP, which are the leftover, they are again goes back to the light reaction, and they are used for the making ATP and the NADPH. So here we can see these three GP, which are the stable product formed from the intermediate product which is unstable. These two G3P joins, and they form the glucose. So this is in short light reaction and dark reaction. In light reaction, with the help of photosystem first and second, the photons are get converted into the ATP and NADPH, and these ATP and NADPH in the dark reaction is utilized and they are converted into the glucose. So here we can see this with the help of this G3P, this glucose is formed, but not all G3P made into the glucose. There is a reason for this, because the Kelvin cycle occur in every stroma in the chloroplast, in every plant cell, and every second of the every day. So there are lots of reaction going on simultaneously. The most of the G3P made during the Kelvin cycle are made into RUBP starting molecule, and they convert this energy ATP into again cycle. So what happens? There are so many G3P, but all the G3P do not get converted into the glucose, because again there is need of the unstable product, that is ADP, and that is again required for the formulation of ATP and NADPH. So if you'll see the summary of the dark reaction in the Kelvin cycle, this Kelvin cycle converts the carbon from carbon dioxide into the glucose in the stroma, and the carbon fixation takes place. So if we see the summary of the photosynthesis, plants are the producer of the biosphere, creating the oxygen and glucose needed for the most organism. Chloroplasts are the site of photosynthesis. We have seen chloroplasts contain the thylakoid where the light reaction takes place, and the light reaction converts the sunlight into ATP and NADPH. This we have seen. And the dark reaction of the Kelvin cycle, this uses the ATP and NADPH to convert carbon dioxide into the sugar. The light reaction and the dark reaction cooperate to convert the light energy into the chemical energy that is housed in the form of glucose. And plant and animal use this energy for the metabolic processes. Now coming to the second part, respiration. Respiration is a process where through cellular respiration, this energy is used for the different metabolic processes. So energy is stored as the organic food in the plant. We have seen the glucose here in the case. And energy is released when this organic food is oxidized during the respiration. And now the potential energy in the food, which is converted into the kinetic energy, and this kinetic energy is utilized for the different metabolic processes. If we see the definition of respiration, it is an intracellular because it happens within the cell, and it is oxidation process in which complex organic substance, and here it is the glucose, are broken down into the simpler substance with stepwise release of energy. So basically this is an intracellular oxidation process where complex substances broke down into the simpler substance to provide the energy, to release the energy which is utilized in the different metabolic processes. So what different raw materials required for this is glucose, which is formed during the process of photosynthesis, and the product is energy that is in the form of ATP, and the byproduct is carbon dioxide. Here is the reaction of the respiration. You can see the glucose which is formed during the process of photosynthesis with the help of enzyme, it get converted into the energy, and the carbon dioxide and the water are the byproducts of it. There are two types of respiration: aerobic respiration and anaerobic respiration. The term itself indicates that the aerobic respiration means it requires the oxygen, and there is a complete oxidation in the presence of oxygen, and it produces the 38 ATP. While in case of anaerobic respiration, there is partial oxidation, not complete oxidation. Therefore, there is only formation of the only 2 ATP, and the anaerobic respiration, the byproduct is ethanol and carbon dioxide. So here is the comparison of aerobic respiration and anaerobic respiration. So if you'll see, this aerobic respiration requires the molecular oxygen, while in case of anaerobic respiration, it does not require the oxygen. Here, the respiratory substrate like glucose, it is fully oxidized. That's why it was creating a 38 ATP. While in case of anaerobic respiration, this respiratory substrate is incompletely or the partially oxidized. End product in case of aerobic respiration are carbon dioxide and water, while in case of anaerobic respiration, the end products are ethyl alcohol, ethanol, or carbon dioxide. So these are some of the differences between the aerobic and anaerobic respiration, and some of the important points are like, requirement of the oxygen and products, and mainly the number of ATP formed at the end. So here we can see the 38 ATP molecule formed from a glucose molecule, while in case of anaerobic respiration, only the 2 ATP molecules are formed from the 1 glucose. So respiratory substrate, these are the carbohydrate, fat, and protein. The important point here to note down is the order of utilization. So first of all, the carbohydrates are utilized, and then if there's no carbohydrate stored, then fats are utilized, and at last the protein are utilized, which is generally under the starvation. So ATP, adenosine triphosphate, it is the energy-rich organic compound which stores the energy. It is the biologically usable form of the energy. So we can, in short, we can say that it is the energy currency for the cell. So energy is released when this ATP molecule is hydrolyzed, and with the hydrolyzation of the single ATP molecule, there is a release of 7.3 kilocalorie energy. And how this energy is stored in the ATP, this ADP, adenosine diphosphate, when combined with the inorganic phosphate in the presence of enzyme, it creates the ATP. So this respiration takes place in the mitochondria. These are the different part of the mitochondria. This is outer membrane, inner membrane. These structures are called as a inner membrane folding structure is called as a cresta, and this liquid portion called as a matrix, and this is the mitochondrial DNA. The function of mitochondria is the synthesis of ATP takes place, and this is the major source of energy. So there are two types of respiration, aerobic and anaerobic respiration. So aerobic respiration there are 4 steps in aerobic respiration. First one is glycolysis, second one is oxidative carboxylation, next is Krebs cycle, and last is ETS, electron transport system. So the glycolysis is nothing but the term itself is saying that the glyco and lysis here, lysis means breakdown and glyco means glucose. The substrate is breaking down here, the glucose. So it is the series of reaction where the glucose molecule made up of 6 carbon is broken down into the 2 molecule of pyruvic acid. This is also called as a EMP pathway because of these scientists Ambren, Merham and Parnas who has discovered this pathway. And this does not require the presence of oxygen, and there is no output of carbon dioxide, and it occurs in the cytoplasm of the cell. So there are total 10 series of reaction and each controlled by a specific enzyme. So this is the glycolysis, and major there are 3 steps here, the activation, cleavage and oxidation. Activation is mainly here the when glucose is activated with the help of ATP, and there is next step is cleavage here, and the product which are formed at the activation stage is cleaved, and last is oxidation process where the oxidation takes place and the energy is released. So these are the different steps which I have explained through the diagram, like cleavage and fragmentation, and this is the oxidation, and the final step is ATP synthesis and the formation of pyruvic acid. If we see the net reaction of the glycolysis, so here the net gain is 8 ATP because 2 ATP is from the NADH2 and 4 ATP are directly from, and also there is a loss of 2 ATP because 2 ATP utilized here in the process. That's why the net gain is 8 ATP. Next process or next step in the aerobic respiration is acetylation. So acetylation is nothing but the conversion of the pyruvic acid, which is the product of the glycolysis. It gets converted into the acetyl coenzyme A with the help of enzyme pyruvic dehydrogenase. Next step is the Krebs cycle. The Krebs cycle is also called as a TCA, citric acid cycle. And this is a stepwise complete oxidation and decarboxylation of pyruvic acid into CO2 and H2O, and it there is a release of energy after this process. This is named after the scientist who has discovered this, Hans Krebs, and who has traced the sequence of all the reaction. This takes place in the matrix of the mitochondria, and it does not consume ATP molecule. So these are the different steps of the Krebs cycle. So this is the Krebs cycle or citric acid cycle. I do not need to remember with the exam point of view. Just you need to remember what is the starting point and what is the end point and how much net gain of ATP is produced. The next step is ETS, electron transport system. This is the final step of aerobic respiration. Here, the most ATP and metabolic water generated in this step And this is located in the inner mitochondrial membrane, Christian oxysome. And individual membrane members are called as electron carriers. An electron from NADH and succinate pass through the ATS to oxygen, which is reduced in the water. So here we can see this NADH pass through the system 1 and the succinate through the 2, and they meet at the ubiquitinous system. And from that, from 3, it will go to cytochrome C, and then 4, and finally this oxygen is it get converted into the water molecule. So this is the flow chart of the same electron transport system, and this is the reaction how the metabolic water formed during this ATS. So this is the most important in the aerobic respiration. We have seen there are formation total 38 ATP. So here we can see during the glycolysis there is a formation of 8 ATP, acetylation 6 ATP, and during the Krebs cycle there is production of 24 ATP. So total 38 ATP are formed during the aerobic respiration. What is the significance of aerobic respiration? So respiration is mainly for the production of the ATP molecule, that is energy. So here the one glucose molecule produces the 38 ATP molecule, and the from the glucose molecule, which consists of 686 kilocalorie, only the 277.4 kilocalorie energy is conserved in the ATP, because the remaining energy is lost as the heat energy. So if you'll see the efficiency of this respiration, it is just 40%. The next or another type of respiration is anaerobic respiration. Anaerobic respiration, because there is an incomplete, because it does not require the oxygen, and there is an incomplete or the partial oxidation of organic food. The organism performing aerobic respiration are called anaerobes, and this in microorganism it is also called as a fermentation, and it is utilized in various commercial process. There is no exchange of gases here, and only 2 ATP molecules are formed. So there are 3 steps in this anaerobic respiration: the glycolysis, decarboxylation, and the reduction. The glycolysis is the same as aerobic respiration here. So here there is, we can say, the glucose with the help of NAD, there is a conversion of glycol, and there is formation of 2 ATP and the 2 NADH. Next step is decarboxylation. In decarboxylation, the pyruvic acid is decarboxylated from acetaldehyde to form acetaldehyde by the enzyme pyruvate decarboxylase. So here we can see, with the help of enzyme pyruvate decarboxylase, this pyruvic acid get converted into the acetaldehyde. That's why this process is called as a decarboxylation. The next step is reduction. So here the acetaldehyde gets got reduced into the ethyl alcohol with the help of enzyme alcohol dehydrogenase. If we see the anaerobic respiration, only the 2 ATP are formed. How the 2 ATPs are formed? Net gain is 2 ATP, because here we can see through the glycolysis there is a formation of 2 ATP and the 2 NADH. But this 2 NADH is required here in the reduction steps. That's why only the 2 ATP is remaining. So the net gain is 2 ATP. In case of aerobic, there are net gain is 38 ATP, and in case of anaerobic, it is 2 ATP. So what is the significance of respiration? So as we have discussed earlier, this is meant for the release of energy. It is for the synthesis of ATP. There is a stepwise release of energy. Energy is required for the growth and development, and this is also for the balance of carbon dioxide and oxygen, and it is required in the various anaerobic respiration in the form of fermentation in different microorganisms required in various commercial process. So the next part of this presentation is factor affecting the photosynthesis. So there are various factors affecting the photosynthesis. It could be divided into the 2 external factors and the internal factors, and we are going to discuss the external factors. So different external factors are like carbon dioxide, light, temperature, water, and DCMU. So the very important law, this Blackman's law of limiting factor, is that what this law said is when a process is conditioned as to its rapidity by a number of separate factors, the rate of process is limited by the piece of the lowest factor. That means if more than one factor affects a chemical process, then its rate will be determined by the factor which is nearest to its minimal value. And it is the factor which directly affects the rate if its quantity is changed. We can understand this with the help of example. We can see that despite the presence of green leaf and optimal light and CO2 condition, the plant may not photosynthesize if the temperature is very low. That means here the temperature is the minimal factor, is the factor which could be a limit, which could limit. So all the factors are required in the optimal amount. If any factor among the all factors required is limit, the process will stop. So if we see the temperature, how the temperature is affecting the photosynthesis. So the rate of photosynthesis increases with rise in temperature, but this is with the range of 6 to 37 degrees centigrade. Above this temperature, there is abrupt fall in rate and the tissue will die at 43 degrees centigrade. So the optimum temperature for the maximum photosynthesis between 20 to 30 degrees centigrade. So in short, the rate of photosynthesis increases with the temperature, but up to a limit, and after that it will there will be a fall because of the dying of the tissue. So here we can see the rate of photosynthesis and temperature. So the maximum is between the 30 to 37 degrees centigrade, and after that the rate of photosynthesis will fall down. The next factor is carbon dioxide. So carbon fixation increases when there is an increase in the carbon dioxide, and the increase in the carbon dioxide increases the rate of photosynthesis. But at the higher concentration of the carbon dioxide may also have the inhibitory effect. So it requires in a optimal quantity. So optimal quantity of carbon dioxide will help in the increase the rate of photosynthesis. So here we can see the photosynthetic response in the C3 and C4. So here we can see clearly the graph is more in case of C4 species because they can accumulate the more C4, more carbon dioxide, and they can convert them into the glucose. The next factor is the light. The quality of light and the intensity of the light, two different factors there. So the wavelength between 400 nm and the 700 nm is the most effective for the photosynthesis. That is also called as PAR, photosynthetically active radiation, because the different rays of light are not absorbed equally by the chlorophyll. And if we see the intensity of the light under low light intensity, the rate of photosynthesis is low, while as the intensity of the light increases, the rate of photosynthesis also increases. If you see the duration, the extent of photosynthesis is more when a plant is exposed to the long period of light, and uninterrupted and continuous photosynthesis for a relatively long period. So you can see the quality of the light, and it is maximum when there is a wavelength between 400 to 700 nm. And if you talk about the intensity, so with the higher intensity, there is a more rate of photosynthesis, and for this more rate of photosynthesis, plants are exposed to long period of light. The next factor is oxygen. The oxygen is essential for the photosynthesis. Photosynthesis does not flex when there is a lack of oxygen, and the energy produced in the oxygen respiration is necessary for the photosynthesis. So high concentration of oxygen inhibits the rate of photosynthesis and it promotes the photorespiration. We are going to see how this oxygen is affecting and what is photorespiration. So water is one of the factor. So here we can see the rate of photosynthesis decreases in the cell which have lost their turgor, and loss of turgor of guard cell close the stomata. That's why it reduces the rate of photosynthesis. That's why the proper maintenance of the turgor pressure with the help of water is important. If there is a loss of water, it will the stomatal cell guard cell will lose their turgor pressure and it will close the stomata and it will ultimately reduce the rate of photosynthesis. So DCMU is a very specific inhibitor of the photosynthesis. So what does it do? It blocks the plastoquinone binding site of photosystem second and prevent the flow of electron from pigment system 2. If it will stop the flow of electron, there will be inhibition of the photosynthesis. The next part is photorespiration. So photorespiration, what is photorespiration? It is nothing but when rubisco join the oxygen instead of carbon dioxide in the first step of Kelvin cycle. It is called as the photorespiration because in the presence of light there is respiration. Generally what happens, we have seen in the Kelvin cycle with the help of enzyme rubisco, this RUVP rubisco 1,5 phosphate, it combines with the carbon dioxide and it converts into the intermediate unstable compound which break down again into the stable compound G3P. But here in case of photorespiration, instead of carbon dioxide, oxygen is utilized. So what is the significance of this photorespiration? So we can see here, and the photorespiration prevents the synthesis of glucose and utilize the plant's ATP. So this is not good for the plant. Why? Because if there is no carbon dioxide is utilized, there is no production of the glucose, and if there is no production of glucose, there is no energy further. And moreover, this also utilizes the plant's ATP. Whatever the ATP or energy possesses with the plant, it utilized by the plant. That's why it is not good for the plant. So here we can see the more CO2 rubisco joins with the CO2P, carbon dioxide to RUVP, and there is a photosynthesis and glucose is produced. But when there is more oxygen, rubisco joins with the oxygen instead of carbon dioxide, and photorespiration occurs and glucose not produced. And what is the main or rationale of photosynthesis is stops due to the photorespiration. So photorespiration is primarily the problem for the water, for the plant, those plants who are under the water stress or other stress. So when plants are under water stress, their stroma closed to prevent the water loss through the transpiration. However, this also limits the gas exchange, and oxygen is still being produced through the light reaction. That's why when there is a closing of stomata, there is no gaseous exchange. When there is no gaseous exchange, there will be no intake of carbon dioxide, which is required for the Kelvin cycle. But here there is a continuous production of oxygen due to the light reaction. That's why there is an increase of oxygen, and that's why instead of combining carbon dioxide, the oxygen is get combined with the RUBP, and it gets there is no production of the glucose. So this is the same what I have explained. So how the plants have evolved to avoid this this photorespiration. So there are different other categories of plant. We call them as a C4 and chem plant. They have evolved in such a way that they avoid the photorespiration, and there are different mechanisms through which these plants, C4 plants and chem plants, avoid the photorespiration, and they survive in these. So C4 plants, in case of C4 plants, C4 plants are those plants where the carbon dioxide is first combined with the 4 carbon product, that is oxaloacetic acid, as compared to the C3 plant where the CO2 is combined with the combined with the 3 carbon product, that is phosphoglyceric acid. And here in the case of chem plant, the chem plant are called as, because crescellation acid metabolism is there in these plants, and their stomata is closed during the day and they are open during the night to avoid the loss of water. So this is it with my presentation. Hope it would help to the listener. Thank you. Thank you so much.
Among the participants having any query regarding the topic, they can ask.
Excuse me, sir. Hello.
Hello. Yes.
Sir, excuse me, sir.
Yes.
Sir, is there any role of oxygen in photosynthesis, sir?
Yes. Of course, there is a role of this whole lecture was there in the oxygen photosynthesis. Definitely, there is a role of oxygen in the photosynthesis. You can see if there is a more of oxygen. I was talking about the photorespiration. If there is more of oxygen, what happens in the Kelvin cycle? Instead of using the carbon dioxide, plant uses the oxygen, and if plant uses the oxygen, there will be no formation of the glucose. So there, the plant should. That's why there are evolvement of the different types of different categories of plant, like C4 plants and the chem plants. So plant has to, or has the balance of oxygen and the CO2, and oxygen is also released as the byproduct in the photosynthesis, which is again utilized in the respiration.
Any other question from participant side? Hello. Sir, can you highlight some of the important points related with the photoperiodism?
Hello.
Aalok, sir.
Yes, sir.
Regarding photoperiodism.
Photoperiodism, so it is mainly regarding the duration of the how the duration and the light of quality of the light affects the flowering in the plant. There are different in photoperiodism. There are different different pigments are involved, like phytochrome A, B, C. So along with this, it has a different scope or different effect on the photosynthesis. How the different spectrum of the light is accepted, or the we can say the different photon is accepted by the different colors in the plant. So if you see the phytochrome, there are different types of the phytochrome A, B, C, and how these phytochrome have the different role in the photoperiodism. And I think so it requires a separate lecture for this.
Okay. Any other questions from the participant sides? Okay, I think there are no any questions.
Hello, sir.
Yes, Lokesh.
Yes,
Sir, what is vernalization? How it play role on flowering, sir?
So vernalization is nothing but the low temperature treatment. When you give the low temperature, there are certain plants it is also the scientists are utilizing the vernalization. Vernalization is nothing but the low temperature treatment, which is generally used for the induction of the flowering.
It's only low temperature required plants only, normal temperature also required. It is only low temperature required plants only, or all plants, sir?
Your voice is not clear, Lokesh.
Sir, it is used only for low temperature required plants or all plants, sir?
No, vernalization is a natural process where generally there are different categories of plant which are flowered only when there is low temperatures given. But what the scientists have done, they have used this natural process for induction of the flowering in those plants where to get the early flowering, in those plants where we getting the late flowering. So generally it is with the lower temperature. And not only this, we can also modify those genes which are controlling the vernalization. And along with the genetics also play an important role in this.
Especially they are used in flowers or all crops they are used at that.
Mainly it is related with the flowering plants.
Okay, sir.
Any other students want to ask? Okay. I think there are no any more questions. I request Mr. Nana Sahib, please conclude the session. Hello. You are not audible, Mr. Nana Sahib. Okay. No issue. I will conclude the session.
Regarding your question, sir, Dr. Prasant sir.
Yes, please.
You were talking about the photoperiodism, how this the long duration or the duration of the light affect the flowering.
Yes.
Like in long day plant, short day plant, how they are responding to the duration of light. Also in that there are, the quality of light where we have to, which category, which spectrum of the light is giving the more rate of photosynthesis. So I think so. If you want, we can have a separate lecture on this if you want.
Yes, sir. Sure.
At some other time.
Okay, sir. Okay. Mr. Nana Sahib. Hello. I think you are not audible. I will conclude the session. On behalf of organizing committee PGI MPKV Rahuri, I am very much thankful to Mr. Aalok sir, as he's elaboratively explained all the information regarding the photosynthesis, respiration, photorespiration, then particularly different cycles which are involved in the metabolic activities of the plants. So he's given the very nice lecture today regarding the general agriculture topic, which is asked during the JRF examination particularly. So I am very much thankful to Aalok sir. Thank you, sir. Thank you very much.
Thank you.
Raj sir, you want to ask?
Thank you. Thanks a lot.
Thank you, sir. Thank you so much. Thank you so much for giving me this wonderful opportunity. And I think so I could have covered this lecture. I mean, more I could have taken more time, and I could have explained in a way slow so that the people can understand it properly. But I also have a lot of things to do, as I've conveyed this message to Dr. Prashant. So
Yes.
I'm really sorry that I could have done it very fast. I mean slow.
No issue. Thank you very much.
We'll do it again sometime.
Thank you, Aalok. See you. Okay. Bye-bye.